I welcome you all on the comstech-oic online certificate course entitled Diagnosis of COVID-19, Sample Collection, RNA Extraction, qPCR, and Data Interpretation. So today we will discuss viral RNA extraction methods, different technicalities which are associated with the diagnosis of SARS-CoV-2 from nasopharyngeal swabs. We at NIV ICCBS are performing COVID-19 diagnostic test at BSL-3 laboratories. So I will discuss a brief about HVAC system, door interlocking system, what is the purpose of anteroom, RNA extraction, both manual and automation, the purpose of the pass box, if any spills happens during the experiment, how we tackle the spill and the waste management. So, the most important question, why RNA extraction is necessary? The simple answer is that we need to find out the main gene of interest that is responsible for causing the disease or the diagnosis of the disease-related gene. In our case, it is the COVID-19. So, it is important to find out the gene of interest, and RNA extraction is the most critical step for performing the diagnostics. In our case, the RT-PCR, because the RT-PCR is the gold standard for the diagnostics of COVID-19. Let's talk about some facts about the RNA. It is extremely hydrophilic molecule with several ionic groups and interchain hydrogen bond. So in order to purify the RNA from DNA, carbohydrates, lipids, and with some structure similar and physical properties, we need to overcome these forces. And these forces are several ionic groups and interchain hydrogen bonds. The polymer of RNA are of different length. It can be short or long. But the problem for the purification of the less or shorter size of the RNA or the smaller stretch of RNA that is less than 100 nucleotide base pair are harder because they do not separate well. In addition, there are also several RNAs present in cells and tissues. So to prevent the degradation during the purification of the RNA or the RNA isolation, it is basically necessary to prevent these RNases because these RNases basically catalyze the cleavage of RNA chains. Viral RNA extraction for SARS-CoV-2. Yesterday, we have learned how to collect the samples, how to take the nasopharyngeal swab, and how to sort the samples. Now, we have a VTM vial containing the nasal swab with the suspected COVID-19 sample. So, these are some critical steps that are used in RNA preparation. We have this VTM, and in this VTM, we have viral RNA, and we need to further extract out the RNA. We will perform all the tests of RNA extraction in BSL-3 facility. So briefly discuss the HVAC system. It is the main component of the BSL-3 facility. It is basically the heating, ventilation, air conditioning system. It actually separates the potentially contaminated laboratory air from the area that are outside the laboratory by maintaining the negative pressure. It has the HEPA filter that provides 99.99% filtration, while the ventilation route is 1, filter exhaust directly through the HVAC system. Now we have French translation.
Merci beaucoup pour votre participation à cette deuxième journée qui, je rappelle, porte sur le diagnostic de COVID-19, la collecte des données, l'extraction de l'ARN, le PCR et l'interprétation des données. Au cours de cette deuxième journée, nous allons aborder le thème qui porte sur l'extraction de l'ARN et les techniques de la PCR pour le diagnostic précis de COVID-19. Donc, à partir d'un échantillon découvert au niveau prélevé au niveau de la cavité nasopharyngienne. La première étape, c'est celle de la collecte des données. Vous avez les laboratoires de biosécurité du niveau 3, et puis maintenant, vous avez l'extraction de l'ADN. Cette partie est présentée par le Dr Saba Farouk, qui est chercheur au PCM à ICCBS, au Centre national de la virologie. Alors, au cours de cette partie, les points suivants seront développés. Les laboratoires de biosécurité niveau 3. Il y aura une partie sur l'antichambre. Vous avez l'extraction, le passage, le déversement et enfin la gestion des déchets. Alors, pourquoi il est important de pouvoir extraire l'ARN? Comme vous le savez tous, lorsque le virus provoque la maladie Corona-19, il se fixe sur les cellules et le petit morceau d'ADN donc le petit morceau est inséré au niveau de la cellule hôte. Et maintenant, il y a la copie, il y a plusieurs virions qui sont formés. Alors, afin de détecter les virus de l'ARN, nous devons être capables d'isoler ces morceaux, ces brins d'ADN de toutes les composantes. Donc, le but ici, c'est de pouvoir isoler uniquement l'ARN. Alors, l'isolement de la haute qualité est une étape très importante, une étape cruciale pour effectuer un diagnostic précis. Voilà les points qui seront développés, puisque vous savez que avec l'ARN, il y a plusieurs molécules, mais il faut parvenir à isoler une seule molécule. Donc, l'isolement de l'ARN est nécessaire pour pouvoir établir un bon diagnostic.
Another component is the door interlock. The door interlock is a double door structure that functions in maintaining negative. It potentially locks up contaminated air and prevents it spreading from outside environment. The interlock system includes the outer door that opens into the anteroom and then into the BSL-3 facility. The doors are controlled by an interlocking system, sensors, and the pin code. These doors are self-closing and cannot be opened at the same time. anteroom, the anteroom must consist of 2 doors for access to the laboratory. Anteroom doors should be interlocked or alarmed, so only one door may be open at a time. It consists of 2 designated areas separate for donning and doffing. The anteroom must be large enough to provide storage of clean gowns, sleep napkins, or uniform that must be donned before entry and removed before leaving the room. The anteroom must provide the space for the log book. Now we are going to start the viral RNA extraction by manual method. Before experimentation, I would like to discuss a brief components or contents which we will use during the RNA extraction. The carrier RNA, which is the poly-A RNA. It has basically 2 functions. It enhances the viral nucleic acid binding to the membrane, and also the addition of this carrier RNA reduces the chances of viral degradation. We will add the carrier RNA into the AVL buffer, and it actually reduces the viral RNA recovery. We will also add the internal extraction control as an indicator for the quality check of the perfect nucleic acid extraction, quality of the samples, and quality of the PCR. During the washing steps, RNA binds to the silica membrane. I'm so sorry for the interruption, and I'm repeating from a few slides from the back because of the poor internet connection. Because of some technical issues and internet connection, I'm repeating some slides back. Please and kindly mute your microphones. Okay, now I am starting from the some facts about the RNA. RNA is the extremely hydrophilic molecule and because of the several ionic group and interchain hydrogen bondings. So, if we want to purify the RNA or extract the RNA for our experimentation or for the diagnosis of any disease, we need to purify this RNA from DNA, from carbohydrates, lipids, and other different structures and molecules. So, we need to overcome these forces, and these forces are several ionic groups and interchain hydrogen bond. RNA polymers are of different length. It may be short or long, but the purification of the short strand of the RNA or the short stretch of the RNA that are of less than 100 nucleotide base pairs are basically hard to purify, and it is difficult to separate the smaller stretch of RNA. In addition, there are several RNases, and these RNases basically degrade or cleave the RNA chain. So in order to prevent the degradation of RNA during the RNA isolation, we need to avoid the presence of RNases. One announcement is that you can also watch live streaming on YouTube by clicking the link on the top left corner. Viral RNA extraction for SARS-CoV-2. We have learned yesterday how to take out the sample from the nasopharyngeal swab, how to sort the sample. Now we have a VTM vial containing the nasal swab of the suspected COVID-19 sample, and we need to further extract out the RNA for the future diagnostic. These are some critical steps for the RNA preparation. We at NIV ICCBS are performing COVID-19 testing at BSL-3 facility. So, I will discuss brief about the HVAC system, which is the major component of the BSL-3 facility. This HVAC system is the heating, ventilation, and air conditioning system. It actually separates the potentially contaminated air from the laboratory and maintain the negative pressure to the adjacent labs. It has the HEPA filter, which provides the 99.99% filtration. Next component is the interlock door. The interlock door is a double door structure that functions in maintaining the negative pressure. And optimum temperature. The interlock system includes the outer door that opens into the anteroom and then BSL-3 facility. The doors are controlled by interlocking sensor and the pin code. These doors are self-closing and cannot be opened at the same time. Next is the anteroom. The anteroom must consist of two doors for access to the laboratory. The doors of anterooms should be interlocked or alarmed, so only one door may be open at a time. It consists of two designated areas separately for donning and doffing. The anteroom must be large enough to provide for storage of clean gowns, lab coats, or uniform that must be donned before entry and removed before leaving the suit. The anteroom must also provide the space for logbook. Now, before moving towards the manual extraction, I would like to discuss the sum component which we will use during the manual extraction. We will add carrier RNA, which is the poly-A RNA, and it serves two purposes. It enhances the binding of the viral nucleic acid to the membrane, and the addition of this carrier RNA actually reduces the viral RNA degradation. We will add the carrier RNA into the AVL buffer, and this addition may lead to reduce the viral RNA recovery. During experimentation, we will also add the internal extraction control as a quality indicator. It indicates the perfect nucleic acid extraction, quality of samples, and quality of PCR. During the washing steps, RNA bind to the silica membrane, and when washed with a buffer of low ionic strength, the DNA is eluted on the column. Now, the French translation.
Merci beaucoup pour avoir participé à cette deuxième séance de notre formation. Alors, il y a eu un petit problème technique, ce qui fait que la récipiendaire a dû commencer. Alors, au cours de cette session, qui je rappelle qui va porter sur l'extraction de l'ARN et les techniques de la PCR pour un diagnostic précis de la COVID-19 à partir d'un échantillon d'échantillon prélevé au niveau de la cavité nasopharyngienne. Alors, quelques faits par rapport à l'ARN. On sait que l'ARN, c'est un polymère avec un groupe ionique. Donc, c'est un polymère qui est extrêmement hydrophile. Alors, ce qui est fait qu'il y ait nécessité de pouvoir les purifier de toutes les autres molécules avec qui elles se trouvent ensemble. Donc, la purification est importante puisque c'est un mélange très complexe. Alors, ce qu'il faut encore retenir par rapport à l'ARN, donc l'ARN comporte il y a des polymères de l'ARN, il y a le polymère qui est long et un autre polymère qui est court. Alors, de manière pratique, il est très difficile de pouvoir séparer l'ARN des différentes composantes et beaucoup plus au niveau du polymère qui est court, qui est difficile à pouvoir isoler. Et là, ensuite, parler de l'extraction de l'ARN. Donc, il y a d'abord la préparation de l'échantillon. Et puis maintenant, on doit faire la lyse cellulaire. Et puis maintenant, on doit avoir les blancs, comme l'ARN. Il faut ensuite utiliser les tampons. Alors, nous sommes maintenant au niveau du système HEVCA, donc qui est un système majeur du système, de PCA, qui a différentes fonctions. Normalement, une fonction comme une fonction filtre, qui peut filtrer jusqu'à 99,89%. Comme aussi fonction, il joue aussi le rôle de maintenir la pression négative Comme vous allez maintenant voir les différentes vidéos, les deux vidéos qui ont été présentées, la première vidéo avait porté sur l'interverrouillage, donc le système d'interverrouillage de la porte. Donc la vidéo 1 qui porte sur le système d'interverrouillage de la porte que vous avez vu. Il y a aussi la deuxième vidéo qui porte sur les porteurs de l'ARN. Alors, il y a deux buts essentiels. C'est de pouvoir bloquer l'interférence, c'est de pouvoir augmenter donc, la pression au niveau de l'acide nucléique. Donc, la membrane au niveau de l'acide nucléique. Alors, maintenant, on va encore repasser les deux vidéos, mais vous allez voir quand les deux vidéos passent, il y a un sous-titrage en français. Donc, à chaque fois qu'il y aura une vidéo, vous allez voir un sous-titrage en langue française.
Now we are going to start the manual RNA extraction. Extraction preparation. First of all, place all the things properly. Equilibrate all the samples and all the buffers at room temperature. Wash buffer AW1 and AW2 are supplied as a concentrate from the company, and it will be reconstituted by using the ethanol, which is indicated on the bottle. If you see any type of precipitation inside the buffer bottle, incubate it at 50 to 60 degrees centigrade. This incubation has no effect on the efficiency of the buffer. Do not heat the buffer after addition of the ethanol. It is advisable to aliquot the buffers to avoid any inconvenience. The necessary equipments for RNA extraction includes Class 2 biosafety cabinets, dedicated pipettes, and tip boxes. First of all, mix the carrier RNA into the AVL buffer. First of all, mix the carrier RNA into the AVL buffer. Initially, calculate the number of samples and the amount of carrier RNA which need to be added into the AVL buffer. Write down the identification number of the related sample on each tube. Now, pipette out 560 microliter of carrier RNA containing AVL buffer into 2 ml of Eppendorf tube. which were already labeled with the identification number of patient sample. The carrier RNA addition buffer to the AVL actually improves the binding of viral RNA to the membrane, especially in case of low viral titer. It limits the possible degradation of viral RNA due to the residual RNase activity. The purpose of the AVL buffer, it is the lysis buffer, which is used for the purifying viral nucleic acid. It is stable at room temperature. Now, after the addition of carrier RNA and AVL, add 140 microliter of the patient's samples to the buffer containing Eppendorf tube. Care should be taken during the addition of the samples into the Eppendorf tube. It is advisable to remove the gloves after addition of the samples. Use RNase-free water as an extraction negative control. Negative control is very crucial during the extraction procedure. It is chiefly used to monitor the contamination or the cross-contamination. Treat the negative control as a sample by taking the 140 microliter of the same volume. Now, vertexing of the sample. This vertexing and pipetting actually produce aerosol of different characteristics and different potential. So the general rule of thumb is to carry out these aerosol producing procedure within the biosafety cabinet. After the addition of negative controls and sample, now add 20 microliter of the internal extraction control. This internal extraction control signal derived from the internal extraction control confirms the success of the extraction step. It can also be used as the quality check or also the presence of any type of inhibitor during the real-time PCR reaction. The control RNA is also co-purified with the sample RNA, and it can be detected as an internal extraction control. Now the short spin. After this, incubate the samples at room temperature. This incubation destroys the RNases and also denature the proteins. After incubation, the next step is the binding. So now the lysis step is completed after the 10 minutes incubation. Now we are at the step of the binding. We need to bind the RNA on the column. So we add 560 microliter of the ethanol and add it into each tube. Ethanol precipitation is actually commonly used technique for concentrating and desalting of the nucleic acids such as RNA and DNA. The basic procedure is that the salt and ethanol are added into the aqueous solution, which actually forces the precipitation of the nucleic acid out from the solution. It also enhances the binding of the RNA to the silica membrane. Now vertexing for the few minutes. After vertexing, we need to apply the samples on the column. But first of all, label the column with the identification code. This is the very important and critical step. After labeling, we have to add 630 microliter of the sample solution containing AVL, carrier RNA, internal control, sample, and ethanol. Into the silica-based membrane column without wetting the rim. Viral RNA actually binds specifically with the column of the silica membrane because of the negative charge. And contaminants are completely removed during the washing step. After addition of 630 microliter of the sample on the column, centrifuge the tubes at 8000 rpm for 1 minute. After centrifugation, you can see the filtrate in the collection tube. After centrifugation, remove the collection tube and place the column in the fresh collection tube. We need to repeat this step because we need to actually complete or finish our lysate. The remaining volume added on the column and again centrifuge at 8000 for 1 minute. Now discard the filtrate, repeat the same procedure and place the collection tube, place the column into the fresh collection tube. Next is the washing step. We are using wash buffer AW1 and AW2. This wash buffer AW1, 500 microliter of wash buffer AW1 is added on the column. These buffers actually contains the cheotropic salts that increase the solubility of the non-polar substances in water. They actually denature the protein because they have the ability to disrupt the hydrophobic interactions, but they do not denature the DNA or RNA. Now discard the filtrate and place the column into the fresh collection tube. This is the ENC, the last one, the extraction negative control. Now the wash buffer 2, which is the AW2, and the addition of AW2 onto the column.
100 की 3 किलो 35 रुपए दिए.
After addition of the AW2, centrifuge at the full speed for 3 minutes. This high-speed centrifugation basically removes all the remaining particles from the column. Now place the column into new collection tube. This is basically the optional step. Okay. Now I'm going back. So place the column into the 2 ml new collection tube. The next is the elution step and the RNA transfer for further PCR processing. Mark new 1.5 ml microcentrifuge tube with the identification code. Discard the collection tube and place the tubes into the 1.5 ml microcentrifuge tube. After placing all the columns into the microcentrifuge tubes, now there is a time for the addition of AVE buffer. It is called as elution buffer. It is used to wash away unbound proteins at first and at a greater concentration. It also releases the desired proteins from the ligand. It is important that elution buffers work quickly without changing the function or activity of the desired protein. This elution buffer is actually made up of RNase-free water with sodium azide. This sodium azide prevents the microbial growth and works as a preservative. Now incubate for 1 minute and centrifuge at 8,000 RPM for 1 minute. Take out all the columns and keep the appender tube containing the RNA in the rack properly labeled. Now place the extracted RNA rack into the Ziploc bag and spray the bag with the disinfectant because this RNA rack in the bag is going out for the RNA RT-PCR through the pass box which is present inside the facility. Now there is a time. No. Sorry for the inconvenience. I'm really sorry for the inconvenience. Now, after the manual RNA extraction, there is a time for the 5 minutes break.
Grazie, sono conoscente la tienesta.
Welcome back. Now, the manual RNA extraction is completed. So, we transfer the RNA for the further processing of RT-PCR through the pass box. What is this pass box? A pass box is a big window-like structure that open to the adjacent laboratory. It acts like a linkage between the BSL-3 and adjacent laboratory. It provides an entry and exit route of samples and small equipment. By providing a passage route, it basically eliminates the need of contamination and constantly open the main door, which would otherwise interfere the directional flow and the negative pressure, hence compromising the containment. The pass box has also interlocking glass door. It is equipped with the ultraviolet light for the surface sterilization. Now, we have some questions. So before moving forward, I would like to address these questions. I would like to address these questions to clarify each and everything. The first question was about the carrier RNA. Either it is commercially available or we can prepare it. So it is commercially available and it is available in the lyophilized form, and we reconstitute it by using the elution buffer. So it is commercially prepared. And someone is asking about the numbering on the tubes. So it is the identification number of the patients, and it is the particular patient identification number which we will use during the processing after till the reporting process. So these are the two questions which I want to address before moving forward. Thank you. So I'm moving towards the RNA extraction by automation. After manual RNA extraction, we are now going to discuss the RNA extraction through the automation. So first of all, I would like to discuss some component which we will use during the extraction by using the automation. Proteinase K. We will add proteinase K. This is the serine protease. And the function of the proteinase K, it actually chew up the viral proteins, capsid, core proteins, and release the RNA. So it will chew up the RNases it come across us. So this proteinase K is very important. It basically provides us the RNA, it releases the RNA, and it chew up the protein, capsid, cores, and also RNases. We will also discuss about the internal control, and we will add internal control in automation as well. And internal control basically extract like a sample, and the combination rule out the inhibitions, PCR inhibition, and other malfunctions. Here in automation, we will use the magnetic beads, and these beads are the silicon dioxide layer which is coated on the magnetic bead, and it can absorb the negative charge of the RNA in order to purify the nucleic acid from the sample. So we are going towards the French translation.
Ok, maintenant nous allons passer à l'extraction de l'ARN par l'automatisation. Alors on va parler des éléments des composantes de cette automatisation. Il y a la protéine kinase K, donc qui est une protéine qui active grandement la sérine, la protéase sérine. Il aide aussi à pouvoir augmenter la vérification des protéines virales, particulièrement au niveau de la capside. Donc, après la protéine kinase, nous allons parler aussi du contrôle positif, donc interne. Donc, le contrôle positif interne est simultanément extrait ou bien amplifié. Donc, cette combinaison aide à pouvoir inhiber les autres malformations. Il y a aussi ce qu'on appelle les bides magnétiques. Donc, c'est le dioxyde de silice. Donc, c'est une plaque avec le dioxyde de silice qui permet d'absorber, qui peut absorber négativement la charge moléculaire dans le but de pouvoir purifier l'acide nucléique des échantillons collectés. OK.
So, unboxing the kit. For automation, RNA extraction, we are using 10 bead 96 well plate kit. First of all, unbox the kit and check if any spill and leakage with the deep well plates. The components of the kit include elution buffers, Lysis buffer tube, plates, magnetic bead plate, washing buffer plate. This is the magnetic bead plate, and we can place the deep well plate, magnetic plate, to settle down all the magnetic particles. Now, before starting the experiment, mark the batch number on the plate. This is the deep well plate. Inside the biosafety cabinet, remove the seal of the Lysis buffer plate. It contains this deep well plate contains the Lysis buffer. After this, add 10 microliter of proteinase K into each well. Like manual extraction, We are adding here 20 microliter of the extraction control into the deep well plate containing the lysis buffer and the proteinase K. So after the addition of internal control proteinase K into the lysis buffer plate, now carefully add the 300 microliter of sample in each well. Care should be taken over here. Now the sample number 2. This is the identification code of the patient sample. Now this deep well plate is completely filled with different types of the samples. After addition of the sample, add negative control. This negative control is RNase free water, and we use RNase free water as a negative control, and we need to extract out this negative control as a sample, and we call it as extraction negative control. After addition of the negative control, we assign some wells for these controls, negative and positive control. Now we are going to add the positive control. So after this, we have extraction negative control, extraction positive control, and different samples in our deep well plate. After adding these extraction negative control, positive control, and all the samples which contain proteinase K and extraction controls, internal extraction controls, we are going to place our deep well plate into machine. First of all, we are putting spin tips very carefully and place done. Then this template move, and now there is a template for placing the lysis buffer in the machine. After lysis buffer, wash buffer 1, wash buffer 2, while placing into the autoplate to the plate holder, make sure that the missing corner of the base faces towards the lower left. On the deep well plate, there is a missing corner, and this missing corner should be fixed on the template present on the machine. So after adding all the plates of lysis buffer, spin tips, wash buffers 1, 2, magnetic bead plates, This is the missing corner of the plate. After adding the elution buffer plate, the machine asks ready and start. It takes around 40 minutes to extract out the RNA, and after completion of the RNA, take out the elution buffer. Check the plate carefully, check the label carefully, check all the deep wells carefully, because sometimes some wells become empty and we need to repeat the samples. Cover the plate immediately. And now transfer the plate through the pass box for further processing. I will again play this video for your understanding. Now we have some questions. So I'm addressing the question. Someone is asking, how can you optimize the purification of short fragment of RNA during precipitation with ethanol? So if you know that your sample contains the short segment of the RNA, there are different types of kits available for this purpose. And for this, you can also use the isopropanol and add it into the sample and spin it for around 40 minutes and then process that sample. Another question is that what type of internal control do you use in this case? So the internal control we are using is basically it depends upon the PCR kit, and it contains the control template RNA that is extracted during the RNA extraction, and the proper probe primer mix contain the primer targeting for this RNA. So it is included into the PCR kit, like internal extraction control. And the next question is how much amount of ethanol was used to concentrate and which concentration? So we are adding 560 microliter of ethanol into the sample, and we are using the absolute molecular grade ethanol. Kindly explain the extraction negative control. Basically, we are using the RNase, DNase free water, which is also provided with the kit. And the next question, is it okay to extract the RNA on bench, not inside the hood? Basically, it is advisable or recommended for RNA extraction to use the biosafety cabinet. As you know that the centrifugation, pipetting and vertexing produces the aerosol. So it is advisable to avoid all these, use the biosafety cabinet. Another question is how much microliter of carrier RNA are used per sample. So we are using 5.6 microliter of carrier RNA. We basically start with calculating or counting the number of sample, and by the counting with number of sample, like for example, we have 12, 15 samples. So we multiply 15 or 10 with 5.6, and we take out the fixed amount of carrier RNA and add into the AVL buffer. Because 5.6 is a very little amount and the chances of error may increase by using this 5.6 microliter RNA. The next is on bench RNA extraction. This is the same question, I think, on bench RNA. Okay. Okay, thank you. Now we have a 5-minute break, and then we will discuss more. Hello, I'll welcome back. I have some questions. I have received some questions. The first question is this: how could you extract RNA without fresh condition? So if you are asking about the RNA or the sample present in the VTM, so it depends upon the VTM. Different types of VTM have different type of sample preservation time, but it is advisable or recommended that you can process your sample within 24 hours or store the sample at 4 degrees centigrade and within 24 hours. Some VTM preserve the sample up to 72 hours. And if the sample is not present in the VTM and it is present in the PBS, so it is advisable to process as soon as possible the sample within 24 hours. The next question is, please name the kit you use for the automatic extraction of RNA. The kit name is the 10-bead RNA extraction kit. And which sample type is used for RNA automation extraction? We are dealing with the Covet samples, nasopharyngeal samples, nasopharyngeal swabs. You can use serum samples. These machines are not fixed these are basically not the closed system. You can use different types of samples and different types of viruses, and we can extract RNA of different types of viruses because these machines have different programs like hepatitis B virus program, hepatitis C virus program, and you can use the nasal swab sample, you can use the serum sample, you can use any type of sample. So principally, most of the samples can be used on these automation machines. But first you have to check that these machines are basically the closed system or the open system. Next is negative and positive control wells are always separate. They are treated as we treat individual samples. Yes, negative and positive control wells are separate, but if we have the negative control well and the very next well is the positive control well, so there is no possibility for cross-contamination if your handling is good. So this is why we are using the negative control to check the cross-contamination. And we usually use the last wells for the extraction positive control and the last 3 to 4 wells for the extraction positive control and extraction negative controls. Is there any worksheet for sample and control addition into the wells for easy identification? It depends. We are using the template or the worksheet, and we basically make a plate map. After sorting of the sample, we make the batches of the sample. Like we make the batch 92 samples batch, or sometimes we make 32 samples batch. So it depends. So after making the batch, we have the particular batch number and the number of samples with the identification code. So we make a proper sheet or proper template, and we follow that template by adding during the addition of the sample. And this template is not shown in the video, but we make this type of template. And it is very easy to make the template if you have the batch with different types of samples. Name of the machine is the 10-bead nanotechnology machine. And how to mention sample ID in the plate. How to mention sample ID in the plate. On the plate, we mention the batch number and the total number of the samples and the date and the district. But We do not mention the sample number. We have the template outside our safety cabinet. We paste the template on the safety cabinet. We only mention the batch number. We know that this batch contains how many samples. The plates are marked by number. Okay, this is the answer actually. Sample which is used for manual extraction, also used for automation extraction. Yes, there is no boundary. There are no boundaries for the samples like this type of sample like we are using with swab samples. So it can be used only for the manual extraction. No, we can. You can use the same sample, same type of sample on both the manual extraction and also for the automated extraction. And basically, it is not the problem that it depends the volume of the sample as well. If you have any spill or if you have the less volume, so we can go through towards the manual extraction. And it also depends that how many samples we have. Like if we have only 12 or 13 samples, so we do not prefer to use the deep well plate, which is based on the 96 well, and all the contents are very expensive. So we prefer that we use manual extraction, but the same samples can be used on the automation machine as well. And sometime we perform one sample on the automation, and after analyzing the results are not clear. So we have to re-extract the RNA and the sample volume is not enough. So we go for the manual extraction because of the sample volume. In manual extraction, we use 140 microliter, and for automation, we use 300 microliter. So it depends upon the optimization, it depends upon your setups, it all depends upon the experience actually. For positive and negative controls, same sample is added into two wells, or in a same well, positive and negative sample is added. No, positive and negative samples are added in the separate well, because positive samples is the positive sample and positive sample carries the viral RNA. So how can we add both the positive and negative control together? Negative control is only RNAs, DNAs, free water. RNAs, DNAs, free water. So we can add negative control separately and we can add positive control separately. Are you talking about the pooling of the samples? I think you are asking about the addition of the sample in a single well. What I can understand from this. You can ask again, please. Basically, if we are talking about the deep well plate, we make the template, we add the sample, like for example, from this deep well plate is of 96 wells. Okay, so we add the sample from 1 to 96, and at the last we basically leave some wells for only negative control, which we called as extraction negative control, and only positive control we can add as extraction positive control. Sample pooling for several sample or sample testing are also basically we can do this as well, and there is an option as well, but sample pooling is not as such recommended, but in this we can pool many samples, many serums, many swab samples, and we can check these samples, and basically in this pooling process what we are doing is that we add different samples in a well, and we process and we extract the RNA. If all the samples are negative, there is no problem. But if any one sample is positive, there is a problem. We cannot say that which sample is positive. So we can take out all the samples and extract their RNAs individually and separately. But I think your question is that you are asking about the wells of the positive and negative control and the separate wells of the positive and negative control. So the next question is automated and manual sample preparation is different? Basically, as mentioned earlier, some sample type is used, same sample type is used, but sample preparation is different, both manual and automated extraction process. Like you also see in this in my videos that in manual extraction we are adding carrier RNA, although we are adding extraction control and all other things, but in automation we are using the proteinase K. So the samples are same, but extraction processes are different. I will discuss the difference between the manual and automated extraction after some time. So next is how long automation take time for extraction. It depends upon the machine. We have different types of machine at an IV. So by using the 10-beat 96 well machine, it takes around 40 minutes. And we have another machine for 24 sample. It takes around 20 minutes for the extraction of the RNA, and 32 machine also take 35 to 40 minutes to extract out the RNA. So it depends upon the machine or the number of samples as well. Can you mention about the reference of SOPs that you have followed? So the samples are processed according to the kit instructions. For RNA extraction, we have the kit instructions that we have to add. Initially, we have to add this and then RNA and then this and this. And if you are talking about the safety SOPs, so we follow the WHO recommendation, CDC recommendation, etc. I'm waiting for more questions. So we are moving forward. So here we are talking about the manual extraction and automated extraction. If we have small number of samples, small setup, less number of samples, manual extraction is recommended. But one thing is that during manual extraction, different manipulations take place with the sample, and there is an increased risk of contamination. As you have seen, different type of steps, different types of washing, centrifugation, vertexing. So it actually increases the risk of contamination. And it is also the time consuming process. And if the person performing the experiment lost his attention, so it will be a big mess, because we are dealing with the patient samples and it is very important to be focused. One key downside to using this method include the potential clogging. We are using the filter silica-based membrane, and because of some sample, because we receive different type of sample. Sometimes we receive samples from the patient which are more viscous, and it is very difficult to take out the samples and to process these samples. So during manual extraction, it is also a potential that the clogging of filter takes place, and because of this clogging, it greatly impacts the RNA yield, and it occurs due to the different type of the sample types. If we are talking about the automated extraction, it has many benefits. Only one benefit. No, this is not the benefit. It is the basically, if we are talking about the cost effectiveness, then manual extraction will be recommended. And if we are talking about the time management and the quality, I think automated extraction has more benefit over the traditional method. And the most important benefit is the consistency. You are not pipette multiple times. You are not using Eppendorf tubes. And because we open multiple times the Eppendorf tubes, we usually change our gloves on after each and after at least 2 steps. Like after one washing, we have to change the glove. After addition of the sample, we have to change the glove. So the most important is the consistency of the nucleic acid, and I think the automated extraction machine has the benefit of it. And if we have the large number of samples, so we need automated extraction machine, because by using the manual extraction, it is very difficult to deal with the 92 samples, because we are making batch of the 92 sample. So if we are doing manual extraction, it is very difficult to perform by the manual extraction. So RNA extraction by automation with the large number of sample is very beneficial. But this is the downside of the RNA automation extraction machine, that if we do not have large number of sample, and if we have a machine and a kit, but if we have 2 or 3 samples, so we go for the manual extraction. Manipulation of the samples and reagent is reduced. This is correct. Like all the samples, all the reagents are already present inside the deep well. We do not need to enter, or we do not need to add any reagent inside the deep well plate. So there is no need of pipetting errors, contamination, and it dramatically reduce or decrease the chance of the contamination. So now the French translation.
Merci beaucoup encore une fois pour la parole. Ici, nous allons énumérer quelques points de différence entre l'extraction manuelle et l'extraction automatisée. Alors, en ce qui concerne l'extraction manuelle, cette méthode est beaucoup plus adaptée pour un petit nombre d'échantillons, généralement dans le but de la recherche. Donc, dans le but de la recherche, comme il y a un petit nombre d'échantillons, alors l'extraction manuelle sera beaucoup plus privilégiée. Par contre, l'extraction automatisée demande beaucoup plus d'échantillons, et c'est une méthode qui est beaucoup plus avantageuse. Il y a plusieurs avantages ici de cette méthode d'extraction automatisée par rapport à l'extraction manuelle. Alors, les plus importants bénéfices de cette méthode d'extraction automatisée est la consistance de l'acide nucléique isolé. Ça, c'est l'avantage le plus probant. Alors, de l'autre côté, en ce qui concerne l'extraction manuelle, l'extraction manuelle demande beaucoup plus de différentes manipulations pour un échantillon qui augmente les risques de contamination. Alors, contrairement à l'extraction automatisée, qui demande moins d'échantillons, et puis ça réduit les risques d'utiliser beaucoup plus d'échantillons au même moment. Donc, ça diminue les chances de pouvoir avoir une contamination croisée. Donc, en ce qui concerne les risques de contamination, c'est beaucoup plus bénéfique d'utiliser une extraction automatisée que l'utilisation d'une extraction automatisée. Alors, dans une extraction manuelle, donc ça demande beaucoup plus de temps. Donc, c'est une méthode qui est exigeante en termes de temps. Et ça demande beaucoup plus d'attention. Contrairement à une extraction, une méthode par l'extraction automatisée qui demande en fait beaucoup moins de temps puisque ça utilise un système qui est automatisé. Alors, en ce qui concerne les désavantages pour une méthode, pour une extraction manuelle, les inconvénients incluent, donc il y a la possibilité d'avoir un colmatage du filtre. Le filtre peut être colmaté, qui peut avoir un impact sur les rendements de l'ARN à extraire. Comme les fiches sont colmatées, il y a beaucoup plus la chance de réduire en fait les rendements de l'extraction, particulièrement pour certains types d'échantillons. Alors, contrairement à l'extraction automatisée, ça demande beaucoup plus une grande quantité, un nombre consistant d'échantillons qui doivent être testés. Voilà un peu les différences entre les deux méthodes d'extraction. Merci beaucoup.
We have some more questions. First is, do you use replica of each patient sample? If yes, then how many replica are used in automation? Usually we do not use replica while we are dealing with the COVID-19 sample because of the cost. If we are using the replica, it will increase the cost. However, if the result is not clear after the PCR and the final result is not clear, or if we have doubt that there is some issue with the sample, like I have already mentioned that some samples come like very viscous samples. So if we analyze some issue with the sample, we extract again sample in duplicate or even in triplicate. But usually we do not use replica in COVID-19 sample. Second, can we use any company's RNA extraction kit, or the automatic RNA extraction is a closed system that require their own RNA extraction kit? We are using 10-bead nanotechnology machine, which is the closed system machine, and we can only use this kit of the 10-bead. Other systems are also available, which give you the option for the open system. You can use different types of kit, and other options are available. But here we are using the 10-bead nanotechnology machine and the 10-bead kit, which is the closed system kit. Next is why we are not adding carrier RNA in automated extraction. Okay, automated extraction, you can see the deep well plate with the magnetic bead. So automated extraction is basically based on the magnetic bead nanoparticles, and these nanoparticles have the positive charge, and RNA bind to on these nanoparticles. If we are talking about the manual extraction, where we use the carrier RNA, this carrier RNA actually enhance the ability of RNA to bind to the silica-based column. So there is a difference. In automation, we are using the magnetic nanobead particles. We added proteinase in automation, and in manual, we added carrier RNA. But this is the same question I have already answered. One question is that how long we can use the mixture of carrier RNA and AVL buffer. We prepare, before starting our experiment, we prepare the carrier RNA and AVL buffer mixture. So it is recommended that we have to use this within 48 hours and place or keep this RNA AVL mixture solution at 2 to 8 degrees centigrade only for 48 hours, not more than 48 hours. So the spill management, another very important area. And basically, this is important in the case of COVID-19 as well. As you all know that these biological spills, if we are dealing with different type of samples and spills occur, these biological spills carries potential risk of exposure to disease. And such spills not only pose a danger to a person who is working, and it also presents the danger for the area, and it contaminate the biosafety cabinet, different type of centrifuges and equipment, even the floor. So there is a very high risk of the spill while we are dealing with different types of the samples, or while we are dealing large number of the samples. So in the biomedical research or diagnostic laboratory, even with the most careful planning, the possibility of incidents always exists. The kind of biospill which is considered as being potentially infectious need to be managed very carefully. And now, as far as COVID-19 or RNA extraction is concerned, if the spill occur during dealing with the samples or during the extraction, the buffers we are using are AVL and AW1. We are talking about the manual extraction. They contain the gonadine salts, and which can form highly reactive compound and combined with the bleach. If liquid containing these buffer spills, clean it immediately with the disinfectant or laboratory detergent and water. If the spill liquid contains infectious agent like the buffer containing the sample, the COVID-19 sample, first of all, clean the area with the detergent and water, and then use 1% volume by volume sodium hypochlorite. Here you can see during the addition of the sample in the deep well plate, there is a spill of the sample. Pick up the sample tube. Now, put the paper towel or the absorbent paper to prevent any aerosol generation. Apply the disinfectant from outward to inward. After the application of the disinfectant, wait for some time. Now decontaminate every item near the spill area. Then apply more disinfectant on the spill area. Once again, wait for some time for disinfectant to work. Wipe out the area one more time. So what happened and what we have to do with this vial, with this spilled vial? Now we have a very less amount of the or less volume of the sample. For this, we go for we check the volume of the sample. If we can, if it the volume is 300 microliter or more than 300 microliter, then we add the sample in the upcoming batch or the next batch to process the sample and the RNA extraction. But because of the spill or due to the spill, now we have a very less sample left. So we go for the manual extraction for the RNA, for the further processing.
Maintenant, nous allons parler des déversements dans l'enceinte de biosécurité. Alors, comme elle indique la vidéo ici, puisque nous sommes dans une zone de biosécurité et parfois on utilise les liquides peuvent se déverser sur la paillasse. Alors, ces liquides augmentent un risque potentiel d'exposition à la maladie suite au déversement des échantillons qui contiennent les liquides, donc qui peuvent, par exemple, dans les cadres espèces, les échantillons qui contiennent les liquides qui contiennent les Covid, donc comme micro-organismes. Et il est important de pouvoir bien gérer cette zone de déversement. Alors, pour pouvoir protéger la personne qui manipule, également aussi avec son environnement. Donc, il est important de pouvoir bien gérer cette zone. Donc, dans un premier temps, il faut d'abord porter, comme mesure de précaution, il faut porter un équipement de protection spécial. Normalement, ici, on parle de l'équipement de protection individuelle approprié et laisser l'armoire de biosécurité allumée pendant le nettoyage des versements. Alors, au cours de la vidéo qui suit, on va voir quelles sont les différentes précautions à prendre. Mais vous allez toujours observer qu'il y a toujours une traduction sous forme de sous-titrage qui est en langue française. Comme vous allez voir après la traduction anglaise, il y a une traduction française. Donc, il faut seulement suivre la vidéo attentivement. Vous allez voir la version française apparaître ici comme sous-titrage. Ok, merci.
Now the waste management. Follow the national guidelines and rules of WHO for the infectious waste treatment. Treat coronavirus waste as highly infectious waste. First of all, separate all the waste sources, and dispose infectious waste into the bin that is suitably colored with the infectious waste bin. Properly handle all the things and transport the things to for the incineration. And for the waste management, use proper PPEs, and which include mask, face shield, gloves, and all the things. If you have the waste of COVID-19 samples, so you already wear the PPEs like Tyvek and all the things. Use safe and environment sound treatment method, and always cut masks and other PPEs, because report that they are being illegally resold. So it is advisable to use, when you use the mask and PPEs, always cut it out, and so it cannot be resold or not be reused. So this is the spill of our sample, and now we have to discard the sample properly in the biohazard bag. So what procedure we have to use? Okay. Now take out the small biohazard bag from the safety cabinet and put into a large biohazard bag. No sharp items should be placed inside this bag because it will puncture the bag. Make the gooseneck and put it in the autoclave. And after autoclave, it will be go for the incineration. Check both the doors should be locked, front doors and the back door. And after checking this, start button press, and the process of initialization of the sterilization is started. And now the lights blink. Now we have 5-minute break. We have some questions. The question is, could you kindly tell me the accuracy percentage of the extraction machine you have been using? So the automated extraction machine is quite accurate. We are using 10-beat extraction machine, and we have never encountered any problem by using that machine with the accuracy. And however, before using 10-beat automation machine, we already did the validation steps multiple times and checks different runs in order to check check the validity and the reproducibility of the results. And after this validation, the machine is used. So the automated extraction machine is quite accurate. Second question is, do you use the DNAs? No, we do not use DNAs. We basically use the tubes and the tips. Our tubes and tips are the DNAs, RNAs free tubes. So in order to avoid any contamination, we use DNAs free tubes and tips. The next question is, in automation extraction, how many type of machine we can use? Which machine is preferred for good and best result? Many machines are available in the market, and many machines are available for auto extraction, including 10-beat, Zsan, and Roche. All these machines are good. And I want to ask, how about the yield of extract, which is better yield between the manual and automated extraction? Both give the 60 microliter sample, and it is very difficult to point out or to give any idea about the yield. If we have one virus or one copy number, it depends upon the results of the PCR. The second is, how could we know that RNA is extracted efficiently? We are using any dye, or are we using any dye and fluorescent? Okay, I have already discussed about the internal extraction control for this reason. We use internal extraction control for the quality check. So if the internal extraction control is extracted properly, process is efficient. Extraction positive control also help in defining the efficiency of the process. When you confirm the result as 100% accurate, and when you confuse about the result. Right now we are talking about the RNA extraction. I think this question is out of the scope of today's topic. This will be addressed when my colleagues will discuss about the RT-PCR and results and data interpretation. They will explain the accuracy of the results. We are now talking about the RNA extraction, and please repeat this question tomorrow. Thank you. If you have more questions, please write to me. Okay, I have some more questions. Why centrifugation is speed and time are different in washing step? Because when we are adding wash buffer 2, AW2, we use high speed between 12,000 to 14,000, because we need to extract out all the volume, all the things, all the contaminants, and we need only the binded RNA. So this is the important thing, that we use the high speed in the second washing, and we use different speed in different washing buffers. Which company kit you select? Why? Basically, we have selected 10-beat extraction kit, and it is the closed system. We are using nasal swab samples over on this 10-bead extraction machine. And the reason is that it is the closed system. That's why we are using this. Any more questions? Please write to me. If not, then we will proceed further. So, after performing all the experiment of RNA extraction, and if any spills occurs and the waste management, the person come out from the laboratory, from the BSL-3 laboratory. So what will the procedure of doffing after RNA extraction? Before coming out of the BSL-3 facility, the outer pair of the glove is removed and discarded. He already removed the outer pair of the gloves inside the BSL-3 facility. Now, the PPEs should be taken off in a designated PPE removal area, doffing area. Disinfect the outer pair of gloves. Remove boot covers and discard them. Now disinfect the outer glove and remove the glove. Again disinfect the inner gloves. Remove face shield. By lifting back of the strap over the head and pull it away carefully. Avoid touching the front surface of the screen. Now again disinfect the inner gloves. Now remove the tie-wick and avoid contact with the outer surface of it. Touch only the inside of the tie-wick when rolling it prior to disposal. Remove the tie-wick in order to roll the whole Tyvek inside out. In order to avoid the contaminated area or the surface. Put all PPEs in a leak-proof biohazard bag. This is for infectious waste. Now again disinfect the gloves. Now break the apron by pulling from the neck and waist. Use the hand again inside out way. Remove it by rolling it inside out. Remove the gloves by inserting the finger inside the inside portion of the glove in order to avoid the touching from the contaminated surface. Disinfect the hands properly. Use fresh pair of gloves after disinfection. Now remove the outer mask by grabbing the bottom tie or elastic strap and discard it. Disinfect the gloves in the same manner. Now remove the surgical mask. N95 and discard it. Now remove and discard the gloves. Take care not to contaminate the bare hands. Sanitize hands properly after the removal of PPE. Now put on the lab coat. Sanitize your hand again. Now the French translation.
Ok, merci beaucoup pour la parole. Après avoir terminé toute manipulation, maintenant vous devez enlever l'équipement de protection individuelle dans une zone qui est adaptée, qu'on appelle les zones d'arrachage. Vous quittez le labo, vous entrez dans une zone d'arrachage. La première chose, vous devez d'abord désinfecter vos mains à l'aide d'un désinfectant. Correctement désinfecter les mains qui contient les gants. Vous allez enlever les dispositifs, les sachets, les plastiques pour la protection des souliers. Vous mettez dans un déchet, dans un bac pour déchets contaminés. Vous allez encore désinfecter les mains avec les désinfectants. Vous allez enlever les prignés G des gants et vous l'enlevez de l'extérieur vers... Vous allez encore à nouveau désinfecter les mains avec les gants qui restent. Vous désinfectez toutes les parties de manière convenante. Vous allez d'abord enlever les premiers masques de protection. Après l'avoir enlevé, vous allez les contaminer, vous allez les décontaminer avec un solvant approprié. Après avoir décontaminé, vous allez essuyer la surface avec un papier hygiénique. Vous désinfectez toutes les surfaces, tous les côtés. Vous allez jeter les papiers et les masques, vous les gardez dans une partie de l'armoire appropriée. De nouveau encore, vous allez désinfecter les mains de manière appropriée, de manière convenable. Après avoir désinfecté les mains, vous allez enlever la première partie, donc l'équipement de protection individuelle. Donc vous l'enlevez à ce que vous ne puissiez toucher autre chose que l'équipement de protection individuelle. Il faut l'enlever de la manière qui est indiquée. Évitez de toucher toutes les autres parties. Donc, vous l'enlevez lentement. Donc, ici, il faut éviter de se précipiter pour ne pas contaminer les autres parties. Alors, vous allez l'enlever en l'enfilant lentement, de haut vers le bas. Vous appuyez pour ouvrir les bacs pour déchets contaminés. Vous allez les jeter. Et à nouveau encore, vous allez encore désinfecter les gants, donc les mains qui portent les gants. Donc vous le faites plusieurs fois. Enfin, vous allez vous retourner. Vous allez enlever la blouse. Vous regardez comment on l'enlève. On ne touche pas toutes les parties. On l'enlève en prenant la précaution. Et vous allez l'enlever en l'enfilant, en l'enroulant encore de haut vers l'en bas. Vous allez les plier. Vous allez encore à nouveau les jeter dans les bacs pour déchets contaminés. Donc les bacs jaunes. Vous allez ensuite enlever les gants, donc les derniers jets de gants, et vous l'enlevez de l'extérieur vers l'intérieur. Vous allez encore les jeter dans les bacs pour déchets toxiques, pour déchets contaminés. À nouveau, vous allez encore désinfecter les mains. Maintenant, ici, vous avez les mains qui sont nues. Vous allez appliquer la solution sur toute la partie de la main. Vous allez ouvrir l'armoire, vous allez encore à nouveau porter les gants, puisque maintenant vous êtes presque prêt pour quitter la zone de retrait, la zone d'arrachage. Vous allez convenablement porter les gants. Vous allez enlever les chapeaux de protection dans les zones de sécurité biologique. Alors, vous désinfectez. Vous allez enlever le premier masque chirurgical. Et puis, vous allez enlever le second masque. Donc, vous évitez de toucher à autre endroit. Vous allez enfin enlever les gants que vous avez portés et vous restez uniquement avec les seconds masques chirurgicaux. Vous allez désinfecter les mains avant de pouvoir quitter la zone d'arrachage. Vous allez porter la blouse médicale, vous fermez l'armoire, vous désinfectez à nouveau les mains. Donc, à chaque opération, il faut toujours désinfecter les mains. Alors, maintenant, vous êtes prêt pour quitter la salle d'arrachage. OK, merci beaucoup.
Thank you. Now we have some questions. The first question is, after RNA extraction, how much time we can store this RNA and what are the conditions? After extraction of RNA, RNA can be stored for months at -20 and for at least a year at -80 degrees centigrade. Next question is, if kit brand change, they are effect of the test? We can only use the kit which is provided with the system, which is provided by the company of the machine, the 10-bead kit. Other kits will not be compatible with this particular machine of the 10-bead nanotechnology. So we are using, because different types of kits have different types of mechanics. Some kits are specific for the hepatitis virus and all the other viruses. So this kit is specific for the particular machine because of the different mechanics. Next, in the pass box, UV light is present. If we pass the sample, is the exposure of UV light create effect on our sample? Basically, this UV light is just for the surface sterilization, as the UV light cannot penetrate if the samples are placed for a very shorter period of time. The samples are taken out within few minutes or few seconds. So this UV light is only for the surface sterilization. It cannot affect the integrity of the sample. The cost of these sophistication is enormous. 3 pairs of PPEs. And yes, the cost is enormous, but it is not more than the safety of our staff. So, any other question? I'm waiting for more questions. One question was based on closed system and open system of the automation machine. So I want to briefly explain what is the open system and what is the closed system. Basically, open system is programmable to use any type of the sample, like it is open system. It including blood samples, serum samples, nasal swabs, oropharyngeal swabs, different types of cell line, etc. RNA, DNA, any type of samples can be used, or any type of samples, RNA from the any type of the samples can be extracted by using the open system. Closed system is not programmable, and I think it is specific for the assay, like some systems are specific for the hepatitis C virus, and some systems are specific nowadays, some systems or some machines are chiefly based on the extraction of SARS-CoV-2. So this is the difference between open and closed systems. Any other questions you can ask? If someone have short hairs, what the specific reason behind is still covering it? Okay. Hair should be covered to avoid any accidental incidents and any exposure to the virus, and it is recommended when we enter into the biosafety laboratory, there are some PPEs which should be taken, and it is recommended to cover the hat or to use the hat cap, and or inside the BSL-3 facility, it is highly recommended to cover. We already use Tyvek inside the BSL-3 facility. Please ask questions. One question is this: how is it impossible that the RNA extraction, it can be stored for a longer period of time, up to years? It is taught to us that after RNA extraction, process the steps as soon as possible, otherwise it start degradation. So it is recommended to use the RNA immediately because the instability and RNA start degrading in the open environment, but it is stable for a longer period of time in RNase free environment. I also had some question regarding the scarf that the person was wearing while entering in the safety lab. It is recommended to use the proper scrubs or proper dress while entering into the biosafety level. And if you are wearing the scarf, you can use that particular scarf only for the facility or only for the biosafety lab, BSL facility or the biosafety lab. Do not use or wear that scarf again. I mean, that person was fully covered, but her scarf wasn't. Basically, inside the BSL-3 facility, usually we wear Tyvek. So there is no option for the exposure of a scarf. Like if we are in the BSL-2 facility, do not wear that scarf and do not go home with that particular scarf. So if you are fully covered but your scarf wasn't, then you have to change your scarf before going home and use that particular scarf only for the BSL facility. Is it okay to use mini pooling during extraction, not during RT-PCR? No, it is not recommended and we are not doing this. Any more questions? What are the problem if RNA molecules are conserved at -20? So it is stored properly, and there is no problem if you store your RNA molecules at -20 for at least 8 to 10 months. But for a longer period of time, you need to store RNA molecules at -80. Please ask more questions. Please clarify how long we can store extracted RNA in -20 and -80. Bit confused. Okay. If you store RNA for a shorter period of time, like for 6 to 7 months, you can store at -20. And for a longer period of time, for a year, you can store it at -80. But as far as our system is concerned, we are dealing with COVID-19 samples, and we only store our RNA samples for 48 hours. And after some time, we re-extract the sample. How about samples after test? If we analyze all the results and we have report all the results, we properly discard all the samples by incineration and by following all the SOPs. Storage before the extraction, if you cannot do the test, can you put it in -4 for 3 to 7 days? If you are talking about the COVID samples, we can process it as soon as possible, all the samples, and we re-extract the samples immediately. How much RNA can extracted by 10-bead kit means yield? No, by using 10-bead kit, the volume of RNA is around 60 microliter. You inserted 10 microliter of proteinase K in well. Is that a standard quality or does it varies according to the kind of samples? We optimize each and every step and every chemical and component. So we optimize it at 10 microliter. We have also checked the 5 microliter volume of proteinase K. It worked well as well. So before incineration, we should autoclave all the disposal. Please correct me. Yes, we have to autoclave all the disposal in the biohazard bag and then we forward it for the incineration. I have some troubleshooting questions regarding the RNA extraction. Discussion, I want to discuss some troubleshootings of the RNA extraction. If the eluted volume is less, after extraction, we have a plate, deep well plate, and if we check, the volume is very less. If it is too much left or not enough less to process the RNA, to process the PCR, we use it. But if it is extremely less, we re-extract the samples or RNA. Magnetic bead is the eluted RNA. So if we are talking about the automation system, sometimes in the elution buffer plate, we have magnetic beads along with the RNA. So what we have to do if we have magnetic bead along with our RNA? So for this, take out the RNA and spin it. After spinning, take out the supernatant and process it for the PCR. This centrifugation basically settle down the magnetic particles and we can easily take out our RNA. And if we are processing all the things and we forgot to add the internal extraction control, which is very necessary for the quality check. So we have to re-extract the sample. Any more questions? Do we need quantification of extracted RNA? In COVID-19 test, we extract RNA and we perform the PCR. And RT-PCR is the qualitative test. Okay, this RT-PCR is the qualitative test. We can easily get the viral copy number. After the results, we can check the viral load and all the things. But if we are not using the RT-PCR and we are dealing with the conventional PCR, we need to quantify the extracted RNA by using nanodrop or spectrophotometer, whatever technique you want to use. Would the re-extraction damage it, re-extraction damage the RNA? I don't think so. Re-extraction cannot damage the RNA. If the RNA is present in the sample, it can re-extract out. if there is no more question, I thank you all for your patience. Do we need to re-extract what? Like if we already extract RNA and if the results are not appropriate, then we re-extract the RNA. And there are some possibility when we re-extract the RNA. If the results are not appropriate and many other factors are present. So then we re-extract the RNA. Otherwise, we do not re-extract the RNA. We are not using the conventional PCR for COVID-19 testing. We are using the RT-PCR. This is the answer in which you asked that you need the protocol of COVID-19 for the conventional PCR. Re-extract performed on sample or the already extracted one. If you are asking about the sample, same sample, once we extract the sample, if the results are not appropriate, then we re-extract the same sample. And if again we have some confusion about the result, we resample the patient, we ask for the resampling. Okay, thank you very much. Goodbye.